I introduced you to different kinds of databases, primary and secondary databases. I introduced you to how a FASTA file looks like. It's a text-based format, what a taxonomy is, and what all information does a typical gene bank record have, um, and the fact that you can have primary databases, you can have secondary databases and hybrid databases. So today I'm going to talk to you about another kind of database. So yes, last time we talked about uh, PFAM, which is a feature-specific database. Feature-centric here being essentially protein features, and protein features start all the way from domains, which is the smallest um, conserved sequence feature, which forms a distinct structural unit in a protein, which goes all the way to a family. And then you can have features like repeats, you can have features like motifs, you have coil domains, and you have disordered regions. The main idea that is unique to all of these features is that these are conserved across different species. So that means evolution is making sure that their sequence is not lost because it must be performing some important function. So now another kind of secondary database, which would be very interesting, which is very interesting and actually very important to use, is organism-centric database. So these databases essentially a database like Ensemble. So if you basically type www.ensemble.org, you will go to the Ensemble genome-centric, organism-centric database for humans. So essentially, instead, if you are working on a specific organism, typically in bioinformatics, in biology, you would be working in an organism-specific manner. So you would typically be taking your, if you would like to look at anything about any sort of biological data, or if we would want to look at specific analysis on this biological data for only humans, then it doesn't make sense for you to go to a primary database or a database which contains all the other organisms. So this is what these ensemble organism-centric databases, that's the problem that they try to solve. Again, they are, because they are secondary databases, so they are based on primary information that is deposited. So these databases are often updated, and you will have different versions of these databases, just like I showed you earlier for the meme suite, where in the meme suite you had different versions of it. They were fixing bugs and things like that. In these organism-centric databases also, they have different versions depending upon what depending upon every update in terms of the human genome, let us say, or the bacterial genome that they are specifically based around. So their main goal is to keep an up-to-date in database of hundreds of other databases that are supporting specific molecular biology and other kinds of information for the organism, and to integrate this all this information in a single access point, right? So that you don't have to go in different you don't have to traverse different databases, say a PFAM database or a UniProt database for protein structure, or the NCBI PubMed for different kinds of things. So how do these databases look like? So Ensemble basically imports genome sequences from consortia. So consortia is basically a group of scientists working together, and make sure that you get consistent information from across many bioinformatics projects, because very often what happens is that when you have multiple researchers working on the same problem, not two researchers are not often uh, presenting the data in the same kind of format, in the same kind of um, don't write, or they don't have a consistent way of representing the same information. So they are typically, ensemble genomes is keeping um, they are keeping information about organism-centric information about important organisms such as human, obviously most important, then mouse, rat, zebrafish. Uh, so mouse, rat, and zebrafish are organisms which are typically used as model systems. So they are, because you can do experiments on them, you cannot do experiments on humans. So typically what they do is, you, if you want to design an experiment that you would like to test a specific uh, test a specific experiment on humans, first you would actually, or you want to make a drug, for example, you would first test the effects on these model systems, and therefore the genome information of these model systems and all these other genomes, genes, all the analysis on based on genomes is also very important, and this is what is stored in Ensemble. So each species basically here is going to have its own Ensemble homepage, and you can find out everything about that species, all the way from the genomes to the final proteins, and everything in between that you can you would wish to know. So here is a short example video of how Ensemble browser looks like. I'm not going to do this in the class. I think it's a 5-minute video. I please recommend all of you to go through it. I am going to ask questions on how to go around in the Ensemble browser. So just to so it's a 15-minute video. So I'm not going to start play this. It's for you to decide. It's for you to look at. So basically, I'm just going to tell you about the key highlights. So what is Ensemble? Ensemble basically, you can do these following things. If you have a gene of interest and you would like to look at homologs in other species, a question that I've asked you earlier, you can just directly go and look for that in Ensemble. If you have a sequence and of your gene of interest, and you want to know what different kinds of mRNA molecules that it's going to make, you can find that in Ensemble. It's a single place. If you want to find out what sequences might be involved in the regulation of your gene of interest, you can actually find that out, such as promoters or enhancers, which are a different kind of promoter, like different kind of way of in which you can control a gene. You can find out how much or which regions of your gene are conserved across species, because it's very important to know the conserved regions are most likely evolutionarily important or functionally important. That's why evolution doesn't want to lose these regions. And you can also know that what kind of mutations are possibly there in your gene of interest or in the location of the genome that you're looking at, because you can then associate these mutations with certain diseases and so on and so forth. So I'll just show you how this typically looks like. If you can all maybe work with me here, please go to the ensemble.org website, everybody. I mean, I think everybody would be on a laptop or on a phone if it's possible. Once you have done this, please raise your hands quickly. So, Satyakam, please, I mean, please raise your hand so that I know how many out of the 8 people actually have done it. So 4 people, I think, have done it. 5. Bhavya, Mayank, Ayush. Yeah. Mayank is left. Okay. Yeah, Mayank, I'm starting. Yeah. All right. So there are a number of ways that you can access the data from Ensemble. You can either directly go to the website, or you can use certain APIs, which are called application programming interfaces, which allow you to pull data directly from the website without logging into the website, let us say for your app. So this is how it looks like. You would have your Ensemble main page. You have these different tabs, which are nothing but different ways of accessing information. On the top right corner, you have a search tab where you can search for anything from any species. On the right side here, you can see the current release version of Ensemble. So here, the snapshot that I have is a release version from April 2020. Can somebody tell me what is the current release number and the version which is put on here?
So
105
105
in
in December 2020.
Right. So yeah, it's okay. 105, and below the versions, you will have the updates that they have made. So they have said that update to Gen code 35 human, Gen code M25 mouse. So these are basically Gen code, just like Ensemble is a aggregator of all information around genomes of different species. Gen code is a consortium which maintains human and mouse genomes, and they also release different versions. So basically, Ensemble updates itself once GenCode releases a new version of the genomes. Then there is another database called Genomad, GNOMAD, Genomad. So it says update of Genomad genome allelic frequencies to version 3. So that basically means, so this uh, database contains the frequency of different mutations that can be occurring inside a locus inside the genome. So they have updated that as well. Three new genomes have been added: three mammals, seven fishes. New genomes added, sorry, three mammals, seven fishes, six birds, right? Four reptiles. So how many new genomes have been added in the last version of Ensemble? In just this the current version, in the current release?
It doesn't say anything
It
in the
doesn't say anything.
updates. Okay.
Okay, so probably
52, sir.
okay.
On 17 December, they added 146 new insect gene. That
Insect
is something
genomes.
that, yes, sir, 146 new insect gene.
Yeah. So basically the idea is, it's like, it's you can imagine that this is a completely different world, right? So just like you have a newspaper telling you what's happening, so these guys are essentially telling you what's happening in the world of genomes. And because this is a curated effort, because the whole idea here is to give you accurate information, which is not just a release of information, but that what information makes sense, right? So sometimes there would be a lag when somebody would be sequencing a genome and when it would be featuring an ensemble, because ensemble is also going to give you lots of other things, as we are going to see. So for example, on the page, you will be having a main search bar where you can specifically select for a species in the search bar, and you can search for a gene like BRCA2. You can search for a genomic locus. So here it is written 5 dot dot 62797383 dash this. So it basically means that you want to go into the fifth chromosome, and you want to zoom into location that is given here after the two colons, right? Or you can search for an RSID here. So RS blah blah RSXXX basically is how you would annotate a mutation in a human genome. Or you can specifically search for a disease such as a coronary heart disease. So it's going to give you all the genomic information which is available around coronary heart diseases. Below this, you can look at all the genomes which are available, and you can specifically go to the genome page. So obviously there is a favorite genomes like human, mouse, and zebrafish. Then at the bottom, you have other tools which is available for you. So for example, you can compare genes across different species. So Ratnesh, what you were asking me, probably this is something that you can do if you have you have a gene, then you can compare that gene across different species, and you can say that, okay, see which species this particular gene is most closest to. You can find SNPs or other variants in the gene of interest. So basically, you ask a gene, or you and you ask what mutations are present in the populations of that species in that gene. You can look at the gene expression in different tissues for your gene of interest. You can retrieve the gene sequence. You can find a data display, so basically some visualization terms, and you can also link your own data with Ensemble. So
Sir, own
again,
data means?
your own data. So for example, you can make your own data and you can upload it to Ensemble, and Ensemble will create a page like this for you.
So, sir, like here we are seeing the release, and even a few changes are happening, like what is 146 new insect genome, then they have a new release. So, like I will add a genome sequence, something like that. So it will be a new release for this.
No, so you can use this framework to analyze your own data. Okay. So for example, you have a new bacterial species that you isolate. You can upload your own data on Ensemble, and it's going to make you available all the other tools that are there on Ensemble. But it's not going to, I mean, it could be a totally, you could have made up a species by randomly typing in ATGC. So they would
Yes,
not
sir.
put the new species until they have actually checked what you have deposited. So it will undergo some peer review, and then it goes on to these websites on these kinds of aggregator. So this is basically a genome aggregator, right? Like you have your Flipkart and Amazon are aggregators for commodities. So this is an aggregator of genomes ensemble essentially. All right. So
So, sir,
you can browse the
again, one more question, sir. Like here in the drop-down menu where the search term, there is a filter for which species you want to search. So, like, I don't think
Yes.
like there is more than 100 species here. So, like, is it the like
Yes.
that in this browser we can search for only these 100 species, or there are more species data also available here which we can search?
So there is two tabs, right? There is a tab, this one, and you have species here, and then there is this all genomes tab, which is here. Okay. So I think in the all genomes tab, you will have more species. Also, these are mostly, if you can see something special about this, ensemble is mostly looking at more popular species. So I'll show you there is something called ensemble genomes, where you have many more species as well. And I'm going to come to that.
Okay, sir. Sir, one more question. Like, why I know that mouse is for and human is like maybe favorite. Like, zebrafish is also something like which is used in research. Something.
Zebrafish is a very important organism because of two reasons. First of all, zebrafish is it grows very fast. So if you want to work with a mouse, let us say you want to create a mouse which is having a mutation in a gene which causes Alzheimer's to the mouse. Okay. Alzheimer's typically is a old age disease, right? So you will have to wait for the mouse to get old, and a mouse typically has an age of 2 to 3 years that it lives. So you will have to wait for 2 years before the mouse is going to start to show some symptoms. All right. Second thing, the act of actually causing mutations in the mouse is or at least used to be quite difficult. So, the way that you search, you see whether you have caused mutations in your organism is that you would do, let's say, you would use a technique such as CRISPR, or earlier people were using other techniques based on homologous recombination, and then you would wait for the mouse to give birth to have progeny, and then you will screen these mice for whether that there is that kind of mutation or not. Now, from pregnancy all the way to delivery, a mouse takes about 1 to 2 months, right? Then the baby grows. So I think it takes about a week's time for the baby to grow enough for you to actually work with it. So now it takes a long time to work with mice. So therefore, you work with organisms which have a much faster life cycle. So for example, people work with zebrafish, which have a complete life cycle of 3 months. They reproduce very quickly. That's one thing. So for another organism, Drosophila, which is a fruit fly, it has a reproduction cycle of a day or two, and it can last just a week, right? So in a week's time, the whole life of an organism is finished. So you can speed up your or experiments extremely fast, and you can use these model organisms. You can manipulate them in much faster way also. And yeah, so if there is a gene which is causing some functional problem, because genes are typically conserved across lots of species, right? So for example, ATP synthetase, the motor which makes ATP, is conserved across almost all the organisms, all the way from bacteria to humans. So if you're looking at
Yes,
some
sir.
mutation in ATP synthetase, you can just might as well work with bacteria first, screen all the mutations, and then you can say, okay, this particular mutation is causing a problem. So let me now go to the next organism. Let me go from a bacteria to, let us say, a zebrafish or a mouse. and let's see whether it causes a problem in these organisms as well. And if it does, then you start to like look at, okay, do these same mutations happen in humans as well? Then you isolate cells from humans, and then you try to see whether these problems exist in humans as well. Okay, that's one reason why you have the existence of model organisms and why you need their. The other thing in terms of zebrafish, which is very interesting, and I mean that's why in I think KSBS there's Professor Shilpi Minocha who works on zebrafish. Um, so zebrafish is very interesting because until uh, a few weeks from its conception of the zebrafish all the way to the growth of the fish is transparent. So when the fish is transparent, so when you will see it, right, you can see all its organs inside. Okay. So for example, if you have to work with mouse's brain, you will actually have to cut the mouse open and then look at the brain of the mouse. Okay. If you want to look at, so that's a problem. So now that means that you have to kill the mouse if you have to look at the brain of the mouse. So that means that you cannot do live
Yes, sir.
experiments. Only recently, what people do, you will find it extremely ghastly, but this is something that happens. They cut a small window in the skull of the mouse and they put a glass window on it. And then on this glass window, they can actually have a window into the brain of the mouse. Right. So this is a complicated process. And very few labs in the world, like I think only a handful of labs in the world can do this. It's a very complicated procedure, right? So now you cut to zebrafish. Zebrafish, for a large part of the like significant part of their lifetime, they are transparent. Okay, so that means if you want to actually look at what's happening inside the brain of the zebrafish, you don't have to have a, you can just look at live what's happening inside the brain. So maybe you can take a gene, you can put a GFP next to the gene, so you can make that gene fluorescent, and you can put the zebrafish under a microscope, and then you can actually look at the firing activity even inside the brain of zebrafish. So zebrafish actually is used for a lot of neural neurobiology, where neural networks are they try to develop neural networks based on how zebrafish is perceiving different impulses. So every model organism has its advantage. Zebrafish's big advantage is that it is a vertebrate, so it has a backbone. So basically, it's much more similar to humans as a fruit fly or an insect model would be, right? And it has a much shorter lifetime, so you can do the experiments much faster, and it is transparent. And if you are interested, you should definitely read about zebrafish. Zebrafish is actually a model organism that comes from India. Obviously, some scientists came to India, they took the zebrafish out of India, and they established the model system. And yeah, so it's found in the Ganges river, in the upper Ganges river. Just like that, there are
Okay.
other model systems which are interesting. Okay, so
Yes, sir.
moving ahead. So
Yes.
ensemble basically allows you to have all these different information, and if you specifically go um, and search for a gene, let us say you go to here on the search bar, you say human and you say you put BRCA2, you would essentially get some information about the gene BRCA2, right? So you can search by the gene identifier, you can search based on some external database, so some UniProt accession, some protein database ID, or a disease name, right? And you will have certain search results. Again, there is a video of how to search things in Ensemble. I again expect you to please go work on this video. This is a homework problem again, and learn how to work with an ensemble. I would expect you to know how this works. There'll be some questions that would be waiting for you for this. Again, this is a quite short video. I'm not going to play it. All right. So when I do BRCA2, how does it quickly look like? So you would have, this is the kind of window that will be opened for you. You will be finding the location of BRCA2. First, it will tell you which version of the human genome you're looking at. So you're looking at GRCH38. So this is the human genome version 38.P13. So this tells you that you are at the 13th chromosome here, and this is the location on the chromosome where this gene is found. This is the name of the gene. This tells you, this tab will tell you what mRNA molecules or what transcripts are made by this gene. Then you have another tab which tells you what mutations are there in this gene, and each mutation can be is given a again an
Correct.
ID, which ID is RS of RS followed by a number. You also have regulation, a tab for regulation. So these are different um, features inside the gene which are responsible for the regulation of this gene. And then you can also submit jobs, like online jobs, like the thing that you did with the meme suite, that you can work with this gene. Yeah, Ratnesh.
Then perhaps now it has been updated and so it looks completely differently. Perhaps your slide is like
It's not old.
uh,
So this is just a specific part of it. This is how it looks like, right?
yes, sir. Yes.
Yeah. So I was just showing you a small part of it. So when you will be searching for, there are like four different regions that you would be having. So I'm going to go to each of these regions one by one. So what I was showing you earlier was this top tab only. Okay. So now let's go to
Yes,
the
sir.
four regions that you have. So you have the top blue region, which is basically telling you about the gene. You have the ID. This is called the ensemble ID. So just like you would have an ID in NCBI, you would have a different or a gene bank, right? You, because you have an entry, it's a database, you need to have an ID for the gene. You would have a description of the gene. You would have synonyms for the gene because the same gene is known by many different ways, many different names, depending upon the function and function of this gene. And different people, whenever they are like discovering genes earlier, they were naming them differently. These functions are typically short forms of these names are typically short forms of the function of the gene, the location of the gene again, and you can get some other information about the gene. When you click on the show transcript table, you get a table of transcripts. This is a summary of the gene in the green here. So you get information about, okay, this gene is a member of the human CDDS set. So this is um, coding CDS set. So this basically tells you, okay, this gene has a protein coding region. This gene, it is written here that this gene protein corresponds to the following UniProt ID. So, it's basically giving you a one-stop location to link this gene to all the other databases, which would be required to tell you something about the gene. At the bottom here, you see um, a chromosome a snapshot of the location of the gene in the chromosome. So this is where the gene is lying. And each of these lines here that you see, so you see couple of solid rectangles and lines connecting these rectangles. So this is basically a transcript. This is how you would, this is the mRNA molecule which is coming out of the genes. The solid rectangles are exons, and the lines which connect the solid rectangles are the introns, so they will eventually be removed. So you can see that BRCA2 makes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, about 12 different kinds of transcripts. So the same gene is making 10 different, potentially is making 10 to 12 different kinds of proteins, because each transcript can potentially make a protein if it has a CDS, as it is marked by a AUG site. On the left side in pink, you have all the other different kinds of things that you can look at. You can look at splice variants. So basically, these are different transcripts that you can get made from the gene. You can have transcript comparison, then you can have sequences, you can have some information about the secondary structure of the gene. Then in one tab, you can do comparative genomics. That is, you can do alignment of this gene across different organisms. You can have a gene tree view. So again, how does this gene relate to other genes and other similar genes and other similar organisms? You can look at orthologs, paralogs. You can look at ensemble protein families. Then you have something about ontologies. So you can have the three ontologies: the molecular function, cellular component, biological process. So it will give you all the ontologies in one place. You can have phenotypes, what is the function of this gene, and if you basically tweak this gene in some way, what is the expected outcome that you would get. Then you can have information about gene variation and all these other things. All right. So basically, again, it's a one-stop shop to know everything about a gene. And now you must be cursing me, saying that why did I make you do all the searching in NCBI? And now there is just one place where you could have got all the answers. So the reason for that is NCBI and all these things were more primary databases where information was disorganized. So you have to search for information for each one of these individual categories. But this is essentially a secondary database where all this information is organized in a single location. So therefore, now you probably understand um, the importance of such databases. All right. So going deeper again. So if you're investigating a specific gene, you will have the gene ID, ensemble ID. If you click on the transcript table, you will get all these different transcripts, and it will tell you whether this particular transcript is going to make a protein or not, and how big that protein is going to be, how long that transcript is, whether that protein is going to actually be protein coding or not, or it's just going to be a potential nonsense mediated decay product. Now, what is nonsense mediated decay is something that you should know in from molecular biology. If you don't know, please go and read about it. It's a process by which the a gene does make a transcript, does make an mRNA molecule, but it does not going to make the protein. It gets degraded in between. And there are these certain other kinds of annotations such as CCDS. So this basically gives you the protein sequence, the amino acid sequence, and UniProt. This basically gives you some three-dimensional structure which might be associated with your transcript or the protein, that molecule that is getting made from these transcripts. Okay. This is the summary area. Again, I've already talked about this. You get some information. You are looking at the specific location of in the genome. So you can see that this is a snapshot of chromosome 13, and these are the coordinates. So it from 32.31 MB all the way to 32.41 MB you're getting. So it's zero point 0.1 MB window you're looking at. And again, you can see that each one of these rectangles are representing exons, and these lines connecting these rectangles are introns. Okay. Now, let us say you go on the left side, you click on sequence. So when you click on sequence, you actually get the sequence of the BRCA2. You can download this sequence as it is. This sequence is highlighted to have exons in a specific color. All the exonic regions are shown here. The regions which are not highlighted are introns, so they will be specifically removed. So exons are shown in red bold lettering and peach highlighting, right? Exons that do not belong to BRCA, so probably this belongs to some other gene. This is upstream region of the gene, right? So this is actually not a part of BRCA2. So remember that. Exons which are alternatively spliced also do not contain the red, are not red. So that basically these means that these are exons which can be alternatively included inside the transcript. When you click on orthologs, you see that for BRCA2, you have 258 species where you have orthologs, right? There are 1:1 orthologs in 22 to 12 species. And then there is a breakup of exactly how these orthologs are distributed across different kinds of organisms. And you can then see they'll basically get a table of what other organisms have this kind of ortholog. Again, you must be cursing me. I asked you the same question where you would had to do a sort of a thorough search of where to find these orthologs for BRCA1, I guess. And but here is a one-stop shop where you can get all this information. But again, that was to show you, to motivate that why secondary databases are required. Okay. So now I'm going to go to another database, which is a hybrid database. This example is UniProt, and why it's important. So UniProt basically keeps an up-to-date comprehensive body of protein information. So the previous one was a secondary database, which was specifically centered around organisms and their genomes. So UniProt is specifically centered around proteins and protein information. And this resource also not only collects information, but also facilitates the discovery and interpretation of the data that you generate when you are working with proteins. So you can do a wide variety of tasks within UniProt. You can compare protein sequences, you can map them to external databases, and you also have a lot of structural information that you can actually put over this your protein of interest. So before I move on to this, I showed you ensemble.org. Just like ensemble.org, you have something called ensemblegenomes.org, and that is something that you can take a look at. And if you have any questions, you can ask me in the next lab. So Ensemble Genomes actually has a lot more other kinds of genomes from bacteria, from a lot of commercially important pests, from fungi, from lots of other organisms. Okay, so UniProt basically is a hybrid database because it has this kind of a structure. So it's a knowledge database. So it takes information from SwissProt. So SwissProt is a manually curated database of protein structures and their IDs. Then you have something called Tremble, which basically is an automated annotation tool. So you have protein structures. If you submit things to Tremble, it's going to automatically annotate it. And then SwissProt people are going to take these automated annotations, are going to check them. And there is this UniProt KB part is built over UniPrac, which basically stores the sequence archives. And then it also interacts, UniPrac also interacts with external sources like EMBL, GeneBank, basically RefSeq. A lot of these are actually from NCBI. UniPrac also, just like you had different versions of the human genome that was being done by GenCode, right? So you had GenCode version 34 on the right side in the release, it was saying. So just like that, UniPrac also creates UniRef datasets. So UniRef 50, UniRef 90, UniRef 100. So you basically have these sequence clusters which can be used depending upon the conservation of a particular protein. And then you are also having proteomes, which is basically sets of all the proteins expressed by an organism. And here you can see this is representing a fruit fly. This is a human, and these are like bacteria. Okay, so just coming back again, it's used to UniProt knowledge base is used to actually access functional information on proteins, and it basically a subset forms entries from the proteome datasets. You have UniProt clusters. It provides clusters of sequences, basically from related species at the protein level, not at the gene level. Earlier in Ensemble, you were seeing when we were talking about orthologs, we were talking about orthologs at the level of genes, not at the level of protein sequence, amino acid sequence. This is more at the level of protein sequence. And then you have UniProt archive or UniPrac, which is basically storing all this information. Okay, so you call this UniProt sort of a hybrid database because there is some part which is manually curated, like in the case of Ensemble, where you have basically it's assured quality, right? You know that all the information, curation, literature that is associated with a specific entry is has a human being who has vetted that information, actually multiple human beings. And then there is also an automated part of this database, which is unreviewed, but it is done by a machine. And this specific part is actually becoming much and much better. Okay, so this is some information about how this entire thing works. Okay, so now we come to some a last bit of information which probably will go towards solving your queries about Go ontologies, is basically the idea of understanding how do we make a good database. So if tomorrow you were to make a good database for biological information or something else, it doesn't matter. There are some principles that we should all look at. So you have to describe data consistently. Okay, that's one important thing for making a good database. Second thing is you should have what's called a minimum information standard. So minimum information standard basically allows you to not describe the same thing in 50 different ways, which is not uh, something which is redundant. So you come up with a specific way of describing information or describing your data, which sticks to a standard. Then in order to achieve this, a database should have controlled vocabularies. That essentially means that the same thing can be described in 50 different ways again, but you need to actually restrict the users to choose from a controlled set of vocabularies to describe a thing. So therefore, you can have a consistent description of data all around. Okay, so I'll now go into each one of these things separately. So first is describing data consistently. So basically for your data to be useful in one set of context, the metadata that you have should actually be done in a proper way. So for example, if you're involved in sequencing samples from an environment, okay, perhaps to understand the biodiversity in different conditions, or to investigate associations between crop yields and differences in different soil flora. So for example, Professor Shilpi Sharma in our department does something like this. It would be useful to know when and where you collected the samples, right? So if you want to compare how does the yield of rice for the same strain of rice is different in Himachal Pradesh versus in Eastern UP, you would like to know exactly where you are collecting the soil from, right? Where you are trying to look at the yields. So you would need standardized descriptors of collecting time, geographic location, and these things can be associated with each time you are collecting samples. So does somebody know any such description of geographical information? So is there a standard format to describe geographical information? In your phones,
In terms of
uh,
coordinates.
yeah, so is there a name for that?
Longitude and latitude.
So there's a longitude, latitude, right? But so there is a specific name for this thing. It's called GIS. It's like global positioning information. So global information system. So if you would, whenever you take your photograph from your phone, I think most of the photos that you would take would actually contain the time, date, and the GIS tag, and you have to actually physically go into the settings of your phone and switch that off if you don't want your photos to be tagged by default. But almost all the photographs actually do contain the GIS information. All right, so now let me give you an example of how this thing works. So let us say that you have same seed samples stored in a seed bank. Now these seeds have been used for doing DNA sequencing or genomic sequencing and proteomics, that is quantifying proteins in these seeds. And these two related experiments need to be compared to each other. Okay, so they are related to each other, and they need to point back to the same biosamples database. Okay, so biosamples database essentially ensures that it collects information about these samples in a standardized format. It's then possible to look at the patterns of gene expression and protein production in this sample and compare them to others and learn about how seeds adapted to a specific environment. Now you can only compare these things if you are comparing apples to apples, oranges to oranges, right? So for each biosample that you have, right, or each sample that you collected, if it will be in a specific biosample format in the biosample database, then based on, let us say, GIS coordinates, based on humidity or whatever metadata that you have collected about the samples, you can actually compare things properly. And once you have large amount of data that is there in the sample, then you, because we are in the age of big data, you can do many different kinds of comparisons that come up with very interesting models where you can start to predict things. Okay, so now when you want to describe data consistently, these are the different things that are required. You need to have a controlled vocabulary and exactly how to report information for this controlled vocabulary. This is called your format. You will have a schema that essentially tells, it's like a form in which this control vocabularies can be used. You will have specific identifiers for this format, which is nothing but pre-decided vocabulary words. And then you will have a ability to distribute this data over a database. Okay, so this is vital for both data and metadata to be represented in a consistent manner. Here again is an example case where, let us say, two groups have been working on figuring out the effect of antidepressants on the gene expression of primary cells in neurons. So primary cell culture basically means these are cells which are extracted from human patients, or like from organism. So these are actually new, like neurons which have not been say differentiated in a petri dish, but they have like neurons coming from the organism. One of them uses a generic name of drugs to describe the experiment. The other one use proprietary names. Okay. Now, because you have isolated the cells from the same tissue using very similar methods, they will have different names for the cell lines and use these in their database submissions. Okay. Now, a computer would think that these two experiments are completely unrelated. And even when a human is searching for one experiment, it would be unlikely to find the other because the name of the drugs are different, and the people will give their own names of the cell lines, right? So, for example, you are working. So, let me take like names of people in the class. So, let us say Dev is working on this experiment, and let's say Mayank is working on the experiment. So, Mayank will name his cells Mayank D dot one, and they would name his cells Dev P dot one, right? And now both of them, one Dev is probably working on a generic version of the drug. So whatever it's the name. So he's actually naming the experiment Dev P dot one, treated with the generic name of the drug. And Mayank will be talking about Mayank D dot one, treated with a proprietary name of the drug. Let us say from a company, let's say Pfizer XXX, right? And they would be working on, let's say, the AFP.1 hydroxychloroquine. Okay, let us say that that was a drug that was being looked at. So now the computer will think, whenever you have submitted this data, people will think, the computer is going to think, even you would think, that these are completely different experiments, right? So that's why it's very important to agree to certain standards to describe data. And therefore databases like EMBL, EBI require researchers to annotate their data with standard controlled vocabularies of information. So this, for the types of metadata that are accepted in international standards, there are many examples, and this is often called minimal information standard. So, for example, a standard notation for longitude and latitude, which is found in GIS notation. So, what do you mean by minimum information standard? So these are guidelines or formats for reporting data derived from specific high throughput methods. Okay, so you can have unstructured data. uh, you basically are transferring unstructured data from journal articles, let's say, into databases, which is the most structured data. And each one of the different kinds of experiments, let us say a microarray experiment, an RNA sequencing experiment, a metabolomic experiment, proteomic experiment, each of them come up with their own formats, own minimum information formats, and that ensure that the information that is stored is enough to compare two experiments, as enough to describe the experiment, and is enough for people to even reproduce the experiment. So, for example, if you are having a publication, in a publication, you have to make sure that if you are having an experiment, that experiment has a proper annotation for this, where the sample is being taken from. So you have to give the species name properly. You cannot just say it is taken from humans. You have to say that this is taken from Homo sapiens, right? Be very specific about it. You have to talk about exactly what were the conditions that what were used for conducting the experiment. What was, even till the batch number of the enzymes or the machine component that you're using. All of this has to be mentioned. If you're generating data, what what time the data was generated, what were the different algorithms used to interpret the data in between, all that has to be mentioned. The whole idea here is that you can compare two experiments, and also you can check whether somebody has done the experiment properly or not, because if all the information is given, then somebody else can actually repeat the experiment and then can check. Is this clear, this part? I mean, this is kind of obvious, but this is something that when you would be looking at papers, you will have a lot of these numbers come up, specifically in the section called materials and methods of a lot of publications. So this is where a lot of this information will be there. So for example, if for a project in the future, you would want to reproduce, or you would want to say, okay, I also want to make this kind of experiment in my project. So in order to understand how to do it, you should go to materials and methods, and there you will be finding these minimum information standard based description of experiments and data, where you will be able to understand exactly how things were conducted or what the experiment was conducted. If this information is not available, then you are in your right to write to the authors or to the publication to provide you with such information. Okay.
So
Now,
such
one very
data
important thing.
were submitted on GitHub, like I see in papers.
GitHub submits not data, no? GitHub is code.
So like I see in many papers that they have like sequences or something like that on GitHub.
It's never on GitHub. GitHub is only code. You can submit code on
Okay,
GitHub
sir.
and you can
I will.
submit intermediate. If there is data
I
on
will check
GitHub,
that.
typically there are, for example, for I can tell you from sequencing data, there is something called Sequence Research Archive, Sequence Read Archive, that's called SRA. For genomics data, for proteomics data, there's something called PRIDE, where this data is submitted. For if you are talking about a table like an Excel sheet, that is often people can share it within the paper only in the form of supplementary data. Or in the case of GitHub, they share code in GitHub. So if you are using some computer program, so you share that code in the form of a GitHub repository, which sometimes has example data sets. And typically the original data set is either coming with the paper itself or it is deposited in a repository, public repository. Um, yeah, so another public repository where often you will see GitHub would be talking to would be Zenodo, Zenodo. Yeah, but GitHub is rarely used for large data deposition because GitHub does not actually give space, that much space to the person who has an account on GitHub, because GitHub is not a data storage. It's not equipped for doing big data storage. Okay, so when I
Yes,
say
sir. Probably the code may be
yeah,
there.
so when I say control vocabularies, I basically meant uh, defining specific words which reduce ambiguity and duplication. So, for example, if you're working with a cell line, so instead of Dev and Mayank, two people doing two people doing experiments on the same cell line, giving a name to their cell line, they will be forced to use controlled words to describe their experiment or their cell line, right? And this basically makes sure that there is less redundancy and ambiguity, and also reduces errors and allows more automation in the process. So here is a quick control vocabulary exercise. So if you can find out 10, there are 10 different ways in which the word human can be described. Can somebody give me a few ways that they can find in this matrix of letters?
The person,
There is
minority,
male, woman, man,
So woman, male, man,
person,
person,
adult.
female,
adult.
adult, right? So all of them actually mean human, right? So you cannot be having all these words. You can only, whenever it's human, you have to actually define it as human. So typically these control vocabularies form non-hierarchical lists. So you have all looked at non-hierarchical lists. For example, when you are asked to choose a language, this is a non-hierarchical list, right? There are no hierarchies here. These are just names. So just like that, when you went to your ensemble database, you could see that it was a non-hierarchical list where each of the species that was sequenced was given, right? Um, so on and so forth. All right, so the control vocabularies can also form hierarchical lists, and these control vocabularies, which are organized in hierarchical lists, are called taxonomies. And taxonomies is one of the oldest form of a classification system. So you would essentially basically a child will be a more specific term from a parent will be a more generic term. And yeah, this is showing you a taxonomy of order of life. So you have life, domain, kingdom, phylum, class, order, family, genus, and species. Just like that, you can have taxonomies, um, which are not hierarchies, but which are networks. And one such example is your Go ontology. So the Go ontology essentially is ontologies are used in informatics, specifically computer science, to represent shared background knowledge. So if you have worked with, I don't know how many people have worked with C++ here. So in C++, there is this whole idea of inheritance of one function from the other function. So they are also forming ontologies. So ontologies essentially is an idea where shared information can form a relationship between two objects, and this relationship can be described in the form of networks. Uh, and these networks essentially are going to be populated again by controlled vocabulary terms to describe something. All right. So hierarchical ontologies take the form of trees specifically, and they become more specific as you go down. So here is an ontology tree. Can you quickly help me complete it? So the top term is organism part. Okay. One of them is heart. All right. So, and the other one is left empty. Can somebody tell me what should come here? Looking at all these things.
The brain.
Brain.
It should, okay. The brain should be here. Okay. Now let's go to heart here. Below the heart, you have the ventricle of the heart, right? And there are three other
Atrium
things.
of heart.
Atrium should come here.
The cardiac ventricle.
Yeah, it should be cardiac ventricle, right? So cardiac ventricle means ventricle of the heart, and ventricle of the heart and cardiac ventricle, they mean the same thing. So that's why they are one over the other. So similarly, you will have cardiac atrium here, and atrium of the heart will be below it. So it will be same here. And then, for example, for brain, you'll have two parts, which is hypothalamus and cerebellum, which is two parts of the brain, right? So this is how you have information that is organized in the form of an ontology. And this is nothing but the same way in which ontologies are described for gene ontologies, right? So in gene ontologies also, you would essentially be having these connections, which would tell you about what a gene is doing. So basically, uh, ontology for molecular life sciences is gene ontology. It describes function, cellular localization, and a process in which a gene product is used. So ontologies are used to describe genes and their products in different public databases. You've already seen those. And this allows you to do what's called gene enrichment analysis, something that I'm going to talk about later in the course. And this is how a typical ontology looks like. So you have molecular function, biological process. So this molecular function, this particular ontology, GO00413, is essentially a molecular process, which is a catalytic activity. Catalytic activity is basically a transferase activity. It is transferase or transferring activity, and the more below you go, the more specific you become. What biological process this is mediating? It's a cellular process and metabolic process. So if you talk about cellular process, it could be a cellular metabolic process. So it derives from both these terms, right? So there is two relationships that are here. Specifically, it could be phosphorus metabolic process. It's a phosphate containing compound, which it's basically working with. Most likely it's a phosphatase or a kinase, which is adding or removing phosphate group, right? Similarly, if you go down this hierarchy, this is essentially working with a cellular macromolecule. It's a protein metabolic process, protein macromolecular modification. So most likely, if you go down, you will find out that this is a kinase that is phosphorylating a protein, right? And if you go up, what was the specific uh, Go ontology? It is protein tyrosine kinase. So it's a enzyme which adds a phosphate group to the tyrosine amino acid in the protein, right? So in order to explain, to describe this, they have come up with this kind of a way. Sometimes it feels redundant, but this is the only way that people have come up so far to organize information in a more consumable format. Okay, so this is how an ontology looks like. And you have three different ontologies. On the top you are less specific, the bottom you go, they are more specific, right? And this basically allows you to explain information about a gene. So there are three ontologies: biological process, molecular function, cellular component. And by looking at these ontologies, you can get a complete picture of exactly what your gene product is doing inside a cell. Okay, you already know what you can do with the ontologies. You can understand what your gene product is doing. You can also create secondary information. If two proteins are having similar kinds of domains, most likely they're going to have similar kind of ontologies. Based on those domains, you can find genes which have similar kinds, genes which are a part of the same process will have similar ontologies. So for example, if you were to look at genes that are going up or genes which are getting upregulated during day, then they would have similar ontologies versus genes which are going up during the night, right? So this is one function that you can achieve with ontologies. You can analyze this in the context of different diseases, in the context of disease, development of an organism, or disease development or disturbance, or basically perturbance. You basically add a drug to the organism and you see how what genes are changing, what ontologies are actually changing when you are perturbing the system or disturbing the system. And you can do this in a very automated way because each gene is now going to have a controlled way in which it is described. So you can actually use your machine learning algorithms to actually do this process in a much more automated fashion. I hope some questions about ontologies were cleared now. If there are any questions, please let me
Yeah.
know.